So the exams are not graded. The TAs have exams of their own, and I have told the TAs that, fingers crossed, I would like to have exams back sometime on Friday. That may not be till afternoon, I don't know at this point. And I'm not even sure if that's possible, but the aim is to get things back by Friday. When exams are available, what I will do is I will send an email out to the class announcing that they're available and announcing where to pick them up. Okay? So they won't be available here, for example. They won't be available in my office, but I will announce where to pick them up. You will need your ID to pick up the exam. Okay? So keep that in mind, and the minute, I can assure you, the minute that they are ready to go, I will put an email out to the class and get them to you. I don't like to have exams out too long before you get a chance to look at them. So I always like to ask, just sort of informally with the class, what did you think of the exam?
Good exam.
Good exam? I'll comment one thing. I have never had an exam where I had fewer questions. There were maybe 10 questions I got on the exam, and that was, for a class of this size, really unusual. So I hope that's a good sign. But you don't have to like my exam. I won't hate you if you don't like my exam. That's fine. Yeah?
Why do you videotape your exams?
Why do I videotape my exams? I find it decreases the looking around factor by a factor of about 100. It does. Yep. I don't do it to be mean, but there's just too many eyes here and not enough of our eyes, and what I have found is that really works very well. That's why I videotape those. And I find most students are honest. I've only had a handful of situations where, in this class, where I've had dishonesty as an issue, and the only students I've ever had with serious dishonesty issues actually were in smaller classes, interestingly enough. Yeah. But it has happened here, and I do have tape evidence when it does happen. So that's why I do it.
Was the Zwitterion question a trick?
Was the Zwitterion question a trick question? No. How many Zwitterions, how many amino acids can't form Zwitterions? None. They all can. That's not trick. You guys should know that, right? Is the answer none a trick? I'm sorry, I don't think so, folks. There are no amino acids. Everything. So think about it. What is a Zwitterion? A molecule that has a net charge of 0. Doesn't every amino acid have a pi? Right? And what's pi? It's the pH at which its charge is 0. Okay. I know I will get some answers on that don't say none. All right? And so I hope that's very few, but that's something that you should know. Yes, Shannon.
What did you have in mind for the last question, the one about the KCAT?
What did I have in mind for the one about the KCAT? Oh, yeah, okay. So the question, and by the way, I will post a key outside my door after we give the exams back. I don't post those now because students get all anxious until they see their exam. So I don't post the key until the exams are back. When the exams go back, there will be a key outside my door. But to answer your question about KCAT, how can you have two apparent KCATs? All right? And the answer is it depends on how you calculate the concentration of the enzyme. If you take Vmax and divide it by the total concentration of enzyme, you will see a reduced KCAT compared to the uninhibited enzyme. Why? Because much of that concentration of enzyme is not active. It's inhibited. However, if you take the inhibited out of it, and you take Vmax and divide it by that modified concentration of enzyme, you will see exactly the same KCAT as if you had an uninhibited enzyme, because you're only measuring the velocity of the uninhibited enzyme when you do that. Okay? So that's the answer to that. That was the thinker question, and I like you to think. I like you to think about the principles that you've hopefully learned in the class. How about length? Was length a factor or not?
Yeah.
How many thought it was too long? Oh, that's relatively small. Okay. I really did try. This is the one exam where time is sometimes a factor. And so I was going to have 3 of the longer answer questions, and I decided not to do that, and I made them shorter. So I had 2. There's trade-offs in all these, as I've told other classes. So one of the things that happens is the fewer the questions I have, the more points each question is worth. And so people are always worried about that. So I have to try to strike a balance. And this balance, I think, works fairly well. I'm glad to hear that there weren't too many, at least, who indicated time problems. Yes, sir.
Have you ever offered alternate formats, like putting out a 5-part exam and saying choose the 4 you want to attack?
Have I ever done alternate formats where I say, okay, pick the ones you want to do? I have done that. They cause logistical problems for grading. And to be honest, I'm not sure that's the best way to test. I don't rule them out, but in my opinion, when I'm giving an exam, I'm sampling. And so I'm sampling your knowledge. And while that sampling can be large and I can do what you recommend there, my concern is, well, what I always see is there's a bias. Okay, everybody decides to do this question over here. And it seems to me that when I have that bias, it suggests that all of the questions aren't equal in difficulty. And so that kind of makes me think, well, maybe that's not the way to go. So, but I have considered it, and I have done it on some occasions. And I won't rule it out. I mean, I always reserve that as an option. But I've shied away from it for that reason. Yeah. Anybody hate the exam? You can say it. That's say I don't. Okay, there you go. You want to say anything?
20-point questions suck.
20-point questions suck. Okay.
No, if you don't know it, that's like 2 letter grades gone.
Yeah, that's why I like having more questions rather than fewer.
Yeah.
Next term, the format of the exam changes in 451. Most people like 451 better because the maximum number of points on a question, I think, is 3. And so we have a lot of shorter answer questions. Yeah. But we're working problems. We have to work through problems, and that's why. If you're going to spend a fair amount of time on something, it should be worth more points. I mean, I could make a 3-point Henderson-Hasselbalch question, but if you spend 20 minutes of your time working on it, that wasn't a good investment of your time, right? Okay. Other comments or questions? Yeah?
You said that time, this exam was an issue. So
This is the one exam where time is sometimes an issue.
okay, so the second midterm is still lecture time.
So the second midterm will be exactly the same format. The final will be exactly the same format. So all the exams are the same format. But what I think happens on the second and third exam is that you've gotten used to Henderson-Hasselbalch format. The working of the problems, which usually is the time limitation, isn't as much of a factor because you've seen how to do these sorts of problems before. And so I rarely, I won't say never, because in a class of this size, I could give an exam where I ask you to sign your name, and people would say it was, I didn't have enough time. But literally, I rarely have much of an issue with the other 2 exams. So, and usually, I'll tell you, when I ask that question to this class, I would say I've seen as many as 2/3 of the hands go up saying, I didn't have enough time. So I was very careful to try to make sure I didn't ask you too many things. But it meant I had to have 20-point problems, because we have to have a 100-point exam. So, yeah, Elizabeth. The extra credit question, okay. I'll answer that, and then I'll go on to other things here. So the extra credit question for hemoglobin, all right. So most people got, the book sort of alluded to the fact that in both cases you had shape changes, that you're changing from R to T or T to R and so forth. And that's true. But the root of that change is the binding of a molecule. And that's the answer. Because in the case of hemoglobin, you're binding a molecule exactly in the same manner that the enzyme is binding a substrate. And so the parallels of those with respect to concentration and so forth, that's really the reason that you see those two curves being essentially the same. Everybody's going, oh. Okay. All right. I hope everybody got how I start my lecture, right? No!
How much leeway are you giving with that?
I said if you said something about starting the exam, we would give credit. Or starting the class, we would give credit. So, okay. Yeah.
I said, how's everybody doing today? Because that's always the next thing that you say.
Oh, that's the second thing I say. So the first thing I say is, okay, folks, let's get started. And I don't do it consciously. I just blurt it out. So, all right. Well, let's turn our attention. Thank you for your feedback. That's not a lot of feedback, but I do appreciate feedback. And I'm always happy to listen to what you have to say about exam formats. And I do take suggestions. The suggestion about having other possible choices is one, as I said, I've done, and I won't rule out. So, but other thoughts or feedback, I'm open to. I very much appreciate that. Okay. Last time I talked about, in some detail, mechanisms by which these S1 proteases worked. And I hope you've had a chance to look through those mechanisms and sort of lay things out. And I know it probably wasn't the first thing you did when you went home after the lecture on Friday, but I think it will pay you to go through and analyze that. Because what you're going to see when you do that is by understanding that mechanism by which S1 proteases work, you will see similarities in mechanisms that other enzymes use to catalyze reactions. And I'm going to talk about some of those today. But before I do that, I want to talk a little bit more about S1 proteases. Because so far, all I've told you about them is that they are a class of enzymes. And they're a class of enzymes that are called S1, are called serine proteases. I'm saying S1 proteases. They're serine proteases. They're called serine proteases because they all use serine in the active site. I said they all have the catalytic triad. But there are some related proteases that have things like that that I want to spend a few minutes talking about today. Okay. One of the things that we think about with the proteases, the serine proteases, is the fact that, as I said, they all have serine, they all have histidine, and they all have aspartic acid as a catalytic triad at their active site. And so one of the questions people have asked is, well, what's the relative importance of each of these in the catalysis of these enzymes? And so using genetic techniques today, it's very easy to alter the genetic code for any of these proteases and change which amino acid is present at any given place. And so doing that, researchers have changed, for example, a serine residue at 221, which is the serine that gives it its name, to an alanine. Or changing the histidine at position 64 to an alanine. Or changing aspartic acid at position 32 to an alanine. Or changing all three to an alanine. And when they do that, and they compare the activity, so this is the log of kcat. So kcat, of course, is a good measure of velocity. And the wild-type enzyme has an activity up here. I note this is a log scale. So this isn't like these are half. This is 1, 2, 3, 4, 5, 6, 7 orders of magnitude, meaning that the wild type is 10 million times more active than the enzyme that has its serine changed to an alanine. Okay? So obviously that serine is a very important residue. You might wonder how in the world it even happens if it doesn't have a serine, and I won't go into that. But this tells us that serine is very, very critical for the catalytic action. 1/10,000,000th as active when that serine is changed to an alanine. What this graph also tells us is that histidine is also very critical, and that's not surprising because, as you saw in that catalytic mechanism, histidine had to pull that proton off of the serine to make the alkoxide ion. If you don't have something that can pull that proton off, then it's a much tougher go. Yes, ma'am?
Is the 221, is that like what amino acid number it is?
That's the number of the amino acid in each case. Yeah. Okay? So without something that can pull that proton off, the enzyme is just as dead in the water as if it didn't have a proton that could be pulled off in the first place. All right? So these are important. When we look at removing the aspartic acid, we still see about 4 or 5 orders of magnitude lower, but this tells us that that aspartic acid residue is not as important as the other two are. The other two are much more important, and that's not totally surprising because the aspartic acid was mainly crowding that histidine. That tells us it's not absolutely essential for its catalysis, although it does play some importance in that. Yes, Neil? So if you're saying, would this be equivalent to what would happen if I put DIPF there? Is that your question?
No, would that first one, would that respond to DIPF?
Would this one respond to DIPF?
The one intake, the first one.
This one?
Yeah.
Would it respond to DIPF? You mean if I made the mutation?
Yeah.
Well, if I made the mutation, I wouldn't have serine there anymore, right? So it would not be possible to treat it with DIPF. So I could only treat something that had serine and expect DIPF to have an effect. Okay? Yes, sir?
Are
Are there studies done on molecules such as this where you could replace the serine, histidine, or aspartic acid with something other than alanine and perhaps increase the function?
Okay, so it's actually a very good question. So his question is, well, what if if I go from serine to alanine, at least they look similar, all right, but they're not chemically similar. What if I change the serine to a threonine, for example? Threonine also has a hydroxyl group. The question is, A, would I see activity, and B, might it even be better? And the answer is, it's all possible, of course, but my prediction would be, in this case, that it would not be, because we're thinking about very precise orientations inside of that active site, and threonine is going to have a slightly different configuration. It's possible it'll be better, but my suspicion is it'll be probably somewhere in here. And his question is also good because when we think about the process that gave rise to these proteases in the first place, they were mutation and selection. Mutation and selection. We think of mutation as mostly being detrimental because most mutations give rise to things that aren't functional, kind of like what these individual ones are here. But some mutations actually give rise to more functional enzymes. That's how enzymes evolved evolutionarily in the first place. And so that tells us something that it's possible it could happen. But histidine to alanine here, that's a pretty big change. What if I put something like, let's say, a tyrosine here that might have a lot of electrons and ability to perhaps influence that proton? That might have an intermediate effect or some other effect. But yeah, make sense? Yes, sir.
Why is it just as ineffective when you replace all three as when you take off one of them?
Okay, so a very good question. Why is this one the same as this? That'd be a good exam question. Why would that be? Yeah, it says that essentially once we disable these, we're pretty much dead in the water. I mean, 1/10,000,000th is there, and that the contributing, these aren't additive effects, but that in fact this is any one of these mutations. Come on. Well, I guess I don't have that. Any one of these mutations alone is enough to knock the enzyme out, essentially. You might say, well, why do you have any activity at all? Okay? And that's also a good question. But remember, we have A, very little activity, and we have the rest of the structure of the enzyme is intact. We have the binding site. We have an environment in there that may be, in addition to the specific amino acid that we see, an environment in there that's favoring, to some extent, these activation and breaking of peptide bonds. So we're focusing on the nucleophilic attack. There are going to be other components to breaking peptide bonds that are still present in the structure of the enzyme. Does that answer your question? Okay, so that's an interesting observation. What I want to do now is show you some other proteases. Okay, so some related proteases. Subtlysin is a protease that's an S1 protease, but there are other proteases that aren't S1 that behave very much like S1, like serine proteases. All right? I keep saying S1. Get that in my head. One of these classes of proteases is known as cysteine proteases. And the cysteine proteases, a good example is papain, comes from papaya fruit. And when we look at this active site, we see something interesting. We don't see a catalytic triad. We see, in this case, a catalytic dyad. And the dyad consists of a cysteine residue adjacent to a histidine. If I were to ask you to predict what you think happens in this catalytic mechanism, what do you suppose is going to happen? How will this enzyme function? I will give you a hint and tell you it also performs a nucleophilic attack, very much like the serine proteases perform a nucleophilic attack. Yes?
The nitrogen on histine withdraws the electron from the sulfhydryl group on cysteine, which is then nucleophilic and attacks the substrate.
Okay, basically, yes, except for you said electron, it's actually removing the proton.
Yeah.
So the histidine is removing a proton off the sulfhydryl of cysteine. That makes a sulfur left behind with extra electrons. Those extra electrons are nucleophilic. They attack the peptide bond, and very much like what we saw, and I'll show you a mechanism in a second, very much like what we saw with the serine proteases, the cysteine proteases work the same way. So let's take a look at that mechanism, and that mechanism is here. It's flipped because now the histidine's on the left instead of being on the right. But we see the histidine. There's exactly that same geometry that we saw with the serine proteases. There's that hydrogen out there for the taking. The binding of the proper substrate to the active site changes the orientation between the histidine and the side chain of the cysteine, making it favorable for those electrons in the histidine ring to pull off that proton from the cysteine side chain. That leaves behind a negatively charged sulfur ion. It's very reactive. It attacks the peptide bond, and exactly all the other things that we saw in the serine proteases happen. We form a covalent intermediate, just like we saw before. We see a fast step, we see a slow step, just like we saw before. So mechanistically, this class of proteases is essentially identical to that of the serine protease, at least for our level of understanding. Yes, Shannon?
When there's an X, does that mean halogen?
When there is an X, that means the rest of the molecule.
Okay.
Remember, these are peptides that we're cutting here, right? Okay. All right. Another class of proteases that is slightly different, but interesting, are called the aspartyl proteases. An example of which is renin. And the aspartyl proteases, at first glance, look somewhat different than the serine proteases, but I hope to show you in the mechanism some similarities that I think you will agree with. But those similarities aren't all the way through like we see with the cysteine proteases. All right? Well, how do these work? First of all, we have, as you can see on the screen, in the active site, we have two aspartic acid residues that are very close to each other. And between them, forming a bridge, is a water molecule. One of the protons of the water molecule is attracted to the negative charge that's on the side chain of the aspartic acid, and it doesn't matter for our purposes here which one. Right? And when we look at the mechanism, we see this. Okay? Here we have one of the aspartic acids on the left, the other aspartic acids on the right. Here's the polypeptide chain that's up here. Okay? And here's the water being held in place. Now, what's happening here, if we think about what happened with the cysteine proteases, excuse me, the cysteine proteases and the serine proteases, we had to abstract a proton to make a reactive molecule. In the case of the serine proteases, we had an alkoxide ion. In the case of the cysteine proteases, we had a negatively charged sulfur ion. What happens with the aspartyl proteases is very similar. We abstract a proton using the electrons of the aspartic acid residue, creating a reactive hydroxide ion. It's got an extra electron. It's negatively charged. And just like we saw in the other two cases, this acts as a nucleophile. The nucleophile attacks the peptide bond just as we saw before. The peptide bond falls apart, and we've broken the polypeptide chain. Now, there is one part of this mechanism I've just described to you that is different from the other two classes of proteases. Does anybody recognize one thing that's going to be very different here?
It flips which residue starts with the water between each one as it switches.
It says it flips between which residue starts the water, and the answer is no. That's not the difference. What's that?
Is it still the same covalent bond being formed?
What covalent bond are we talking about?
In
You're on the right track. That's why I'm asking you.
the intermediate.
Okay, so she's right. If we think about for example, the serine proteases, we made an alkoxide ion, right? We had that oxygen atom that had an extra electron. It attacked the carbonyl group. That caused one part to fall off, and what happened to the oxygen? It became covalently bound to the other chain. That chain was therefore stuck at the enzyme. That oxygen was attached to the rest of the enzyme, right? Do you see this water being attached to the rest of the enzyme? No. So we're not going to see a fast step and a slow step with this. Once we break the bond, we break the bond. There's nothing holding it to the enzyme such that the other chain has to be released. That's what made the slow step in the serine proteases. Everybody understand that? In the case of the serine and the cysteine proteases, the nucleophile was physically attached to the enzyme. The oxygen was the side chain of a serine. The sulfur was the side chain of a cysteine. Those are covalently attached to the enzyme. So when they become covalently attached to half of the polypeptide chain, that half of the polypeptide chain has to get released. Here, water is not attached to anything. When water attacks that carbonyl group, the bond breaks. It's free. One step. Bang. Everybody got that? No? Questions?
So is it all just fast? Is it all very fast?
I won't say it's fast or faster because remember we have to still get that proton off. So I'm not trying to compare speeds. I'm just saying we won't have two steps, a fast step and a slow step. We won't have that with an aspartyl protease. Yes, sir?
Does this kind of reset itself by just stealing the hydrogen off the, sorry, I'm not saying this very well, the right side
Yeah.
carboxyl?
Yeah.
Is that how it resets itself, or does it actually go, does the charge go back to that side?
Yeah, your question is a very good one. How does the enzyme reset itself? And if we think about the water that was added, one of the protons gets grabbed here, and the hydroxyl does the attack. But to break a peptide bond, we have to add both an OH and an H. And so that H can either come from here, that H can come from here, and then the enzyme to reset itself is going to have to get back to its original state. So if it comes from here, then it's a very simple matter. We've just gone back to the original place that we were at. Okay? Okay. Let's move on to the third class of protease, the third class, and these are called metalloproteases. Now, we're getting further and further away from serine proteases, but I hope you see a common theme. An example of a metalloprotease is thermolysin. These enzymes derive their name by virtue of the fact that they use a metal ion as their way of holding onto a water molecule. We'll see this theme come up also. A metal ion to hold onto a water molecule. We saw in the aspartyl proteases that the water was in place, and the water had to get positioned right there. All right? The water played an important role in that catalytic process. The metalloproteases have a means of holding water there. That actually could make the enzyme more efficient because in the aspartyl proteases, that water can be bouncing around. That's why I didn't say they were faster. But in the case of the metalloproteases, we've got something that's going to hold on to the water. Well, the most common ion that's actually used is zinc. And I'm going to show you that mechanism here. In the case of metalloproteases, we're looking at the active site. And we remember that if we're going to break that peptide bond, we have to have a nucleophile. And that nucleophile is coming from water. We're going to make a reactive hydroxyl group, just as we did for the aspartyl protease. But the way in which we make it is slightly different. First, water is bound by this zinc ion. The zinc, you'll notice, is positively charged. The positive charge of the zinc is attracted to the negative, or the relatively negative charge, partial charge, on the oxygen of the water. That interaction helps to hold water at the right place. And then, and this is going to vary from one enzyme to another, there is a side chain that will help to remove a proton from water. That might be a histidine. That might be another like lysine or arginine or something else capable of pulling a proton. It's listed on there as a B for base. So some other side chain is going to help in the removal of that proton from the water. And again, once we've removed the proton, we've got a hydroxide behind that is very electron rich, attacks the peptide bond, and bang, we're off and running. Is this going to have a fast step and a slow step, or just a fast step?
Two-step.
Fast and slow? Okay, what does it take to have a fast and slow? What's the slow step in a serine protease? releasing the polypeptide chain from the enzyme, right? Do you see the polypeptide chain getting attached to the enzyme anywhere here? No. This only has one. This doesn't have a fast and a slow. This does not have a fast and a slow because there's no covalent intermediate with the enzyme. The water, again, is the attacker. The hydroxide, in this case, is the attacker. Okay. Yes?
Is the water actually bonded to the zinc, or is it positive, negative?
The water is attracted to the zinc. It's not a covalent bond of any sort, no. Okay, so these are partial ionic interactions, not unlike a hydrogen bond, except for it's not a hydrogen that's involved. It's an oxygen. Okay? Okay, so hopefully what you see in that is a common theme. First of all, every single protease that I've shown you so far has a nucleophile. Every one. That nucleophile is what attacks the carbonyl group, and it's the attack on that carbonyl group that results in the breakage of the peptide bond. Okay? That common theme goes through all of them. The only ways in which these enzymes that I've described to you so far differ is the means by which they generate the nucleophile and what the nucleophile itself is. That's the only ways in which they differ. But the basic mechanisms are the same. We create a nucleophile, the nucleophile attacks the carbonyl group, the peptide bond breaks, and the pieces go their way. Okay. Well, this business of creating nucleophiles is not unique to proteases. There are other enzymes that use nucleophiles and generation of nucleophiles in their catalytic mechanisms. One of these is an enzyme we've been talking about some already. That's the carbonic anhydrase, and that's right here. Okay. You guys look very tired. Would you like a joke? Would you like to stretch before the joke? Okay, so let's stretch. Yes, sir. Oh, yeah, get Norton out of there. Okay. So this is my magic genie joke. All right, my magic genie joke. This guy's walking along down the street, kind of bummed, kicks a bottle, and all of a sudden realizes, "Whoa, what's that?" He grabs it, wipes the dust off of it, and of course in the process polishes this thing, and out pops this magic genie. Yeah. And the magic genie says, "Oh, master, thank you. I will grant you the 3 wishes, etc." And the guy says, "Oh, this is really great." He says, "First of all, what do I want? I guess, I want, $1 billion, so that I can be a very rich man." And poof, the certificate appears in his hands, and it says he has $1 billion in a Swiss bank account. This is really good, right? Second wish, he says, "I'd really like to be a very powerful guy." Okay? And poof, the certificate appears in his hands, and he's the president of Apple. Or Microsoft. It doesn't really matter. This is not a computer joke. Depends on where you lean, I suppose. This is really awesome. I'm really a powerful guy. He says, third wish. Well, let's see. I've got money. I've got power. I want every woman to love me. And poof, he turns into a box of chocolates. And you thought it was going to be a dirty joke, didn't you? Okay. That's a dumb joke, I know. But it always gets a big laugh, so. I hate telling jokes about one sex or the other. But, All right. Carbonic anhydrase. So, you've heard about carbonic anhydrase already. I hope I've convinced you it's a pretty amazing enzyme. Carbonic anhydrase, you recall, was the enzyme I said when we first started talking about enzymes that had a kcat of 1,000,000 per second. It can catalyze the conversion of 1,000,000 molecules of substrate into product per second per enzyme. Remarkable thing. Okay? Here's the reaction that it catalyzes. Okay? We see carbon dioxide plus water going to carbonic acid. Carbonic acid can then ionize and form bicarbonate. And that can happen with incredible rapidity under the right conditions. Okay? Under the right conditions. Now, what does that mean? Well, there's something very odd that happens with this enzyme. Most enzymes have a fairly narrow pH range where they work that's ideal. And most of those ranges are set pretty much to correspond to the physiological environment in which we find them in the body. Most of our body tissues are at a pH of 7 to 7.4. Okay? Blood, for example, is about 7.4. When we look at the activity of carbonic anhydrase, and by the way, you're seeing kcat changing here. Kcat will be constant for a given set of concentrations. If we change the concentrations, kcat itself will change. We look at the kcat of this enzyme, and we discover something very odd. We see, not surprisingly, at a low pH, it's fairly low. We get to about pH 7, and we're looking maybe 500,000 per second. But this guy tops out at about pH 9, where we get up to 1,000,000 per second. Why is this enzyme odd in this respect? Why is its kcat so high at such a high pH that it really doesn't encounter in the body? Okay? Well, the reason that this is the case is it gives us a clue into a very important consideration in how the enzyme works. All right? And so that's what I want to show you next. And it's going to relate to the things I've been talking about. All right. During the catalytic process inside of this enzyme, we see something that happened very much like what was happening in the metalloproteases. That is, we have a zinc ion, and that zinc ion functions to hold water so that we can create a nucleophile. It functions to hold water so we can create a nucleophile. The rate-limiting step in the catalytic action of this enzyme, like that of the proteases that you saw before, is the rate of formation of the nucleophile. Okay? The rate of formation of the nucleophile. The faster the nucleophile can form, or the easier the nucleophile can form, the faster the enzyme is going to be. If we go to pH 9, we could imagine that it's going to be a heck of a lot easier to remove protons off of water than if we're at pH 7. We have more basic conditions. Water is going to give up its protons more readily. And so at pH 9, we see that this enzyme is far more active than it is at pH 7, indicating that a very, very important step is the removal of that proton. That proton can come off many ways, but if the buffer is helping that proton to come off, it does even better. Does that make sense? I see a lot of nods. Okay, good. Now, my next question is, do you suppose if we kept raising the pH, it would get faster and faster and faster? I see no's. As they say, it's a trick question. I know Ahern, right? The answer being no, why? Well, the nature of the enzyme. Okay? So at pH 9, the enzyme is still holding its shape enough that it's actually able to continue catalysis, but we get above that, we're going to see the enzyme just drop off precipitously. Okay, and so that's a very interesting step. This actually shows the mechanism, and again, it's nothing like you haven't already seen. We see the water being bound. We see something pulling off that proton, whether it is just ionization of the water itself, which will happen in water to a limited extent, or it's like a side chain of a histidine or a lysine or an arginine that is pulling that off. Once we've got the nucleophile created, the nucleophile attacks carbon dioxide as it diffuses into the active site, and that actually creates this, in this case, bicarbonate ion that is released. Okay? So it's the formation of the nucleophile, just as we've been seeing all along, that is the critical step in this process. Would you predict that aspartyl proteases might work better if we raise the pH on them? You can stick your neck out. I won't chop it off. I hear a no. Do I hear any yeses? What do aspartyl proteases have to do, folks? All right, what about the metalloproteases? They both use water. How about metalloproteases? Would they work better at a higher pH? Probably would. They probably will work a little better at a higher pH. And what's going to determine if they work better at a higher pH or not? The stability of the enzyme structure. That's going to be the only limitation. If the enzyme structure is stable to pH 10, the enzyme will be way better at 10 than it is at 7. Because, again, it's easier to make that nucleophile at pH 10 than it is at pH 7. Okay. Yes, sir?
So that being a consideration, is there any movement like the biotech industry to create man-made enzymes by taking a natural enzyme, sticking in a bunch of cysteines and cross-linking it back and forth like crazy and making it stable at a higher pH?
You should work in the biotech industry, sir. As a matter of fact, yes, there's a lot of interest in manipulating enzymes to, in fact, increase their activity and increase their efficiency. In the case of carbonic anhydrase, though, remember that what's limiting them is actually diffusing it into the active site. It's probably not going to get much better than that million, even if we were to improve that. But for a metalloprotease, it might very well be useful, because now we can stabilize the enzyme with disulfide bonds. We could use it at a higher pH. That might very well be a strategy. You bet. Okay. Good. All right. We're slithering along through this. Let's spend the last 5 or 6 minutes talking about restriction enzymes. We'll see some similarities we saw before. So you really I hope you're starting to see the themes now. Restriction enzymes are enzymes that many of you have used if you've ever worked in a lab that does DNA work. And restriction enzymes catalyze the breakage of DNA double strands at specific nucleotide sequences. Restriction enzymes, they're also called restriction endonucleases, and again, I'll take either one, that's fine. They catalyze the breakage of DNA double strands at specific nucleotide sequences. Now, that's what they do. That's not how they do it. How did they do it? Well, I will tell you that they hydrolyze them. They use water to break the bond. And that should give you some hint about the mechanism that they use, and you can start seeing the wheels turn, thinking, I'll wager that they're going to make an activated water molecule in some way, like taking a proton off, making a nucleophile. Nucleophile is going to attack, and that attack is going to result in breakage of a bond, and you would be exactly correct. Okay? You would be exactly correct. Please turn your phone off, whoever has it on. Okay? Now, let's take a little bit closer look at this in terms of how they operate. Okay? Actually, that's not a very good figure. All right. One of the things we see in restriction enzymes is that they all require magnesium for their action. They all require magnesium. And magnesium, like zinc, is a divalent cation. And one of the things that magnesium helps to do is it does help to position the water so that it can lose a proton and make an attack on, in this case, a phosphodiester bond. We're not breaking peptide bonds, obviously. We're breaking phosphodiester bonds because those are the bonds between adjacent nucleotides in a DNA molecule. Now, I'll show you something that's really cool and interesting. Actually, I have to explain it to you because it doesn't show it very well. What I just did, the wrong one. Let's take a look at this enzyme. There are many different restriction enzymes. This one's called EcoR5. EcoRV, right? EcoR5 recognizes the structure, the sequence GAT, ATC, and it cuts right in the middle of it. Now, if you look at this carefully, you'll see that on the top strand we go GAT, or ATC. On the bottom strand we go GAT, ATC. These are what are called symmetric sequences. And these symmetric sequences are very common features of sites that restriction endonucleases recognize. It means that we can cut right in the middle of this guy, and we've cut both of them. Right? We'll just cut this guy right in the middle. Not all of them work in the middle, but this particular guy works in the middle. All right? Now, I want to explain to you just physically how a restriction enzyme works, and then I'll show you very briefly a little bit of mechanism. All right? A restriction enzyme is a protein. That protein grabs a hold of DNA. Okay? So many proteins will grab a hold of DNA. DNA is a negatively charged molecule. We would expect that to grab a hold of DNA, perhaps positive or neutral, we certainly wouldn't expect the protein to be negatively charged because it wouldn't interact very well with DNA. The enzyme grabs a hold of a DNA molecule, and what it does is it literally slides down the DNA molecule. Now, most of the time it's going down that trip down the DNA molecule, it does not encounter the sequence GAT-ATC. In fact, that sequence will occur randomly only about once every 4,000 residues. So a lot of the time it's just floating along here, sliding itself down the DNA molecule, and then all of a sudden in the binding site of the enzyme, GAT-ATC is there. It's found the right site. Okay? What happened in the serine proteases or in any of the proteases or in any of the enzymes when the proper substrate bound? What happened physically inside of those enzymes? Shape change, right? We saw a shape change that happened in those. Those very tiny shape changes caused the enzyme to have all of its properties. In the case of the serine protease, that shape change resulted in the creation of the alkoxide ion. In the case of the restriction endonuclease, that shape change is more dramatic. What it does is it actually causes a bend to occur in the DNA. So we think of the DNA molecule as being straight and linear, but when the enzyme is bound to that proper site, the enzyme goes, oh, whoa, and it bends. The DNA molecule is physically bent at that point. It's physically bent. Now, that bending turns out to be critical for the catalytic action. Okay? I've got to keep going up there. Now, we can see that bending happening right here. And when we analyze the structure that's present at that bending, what we see, first of all, is that there's a nice little water molecule that gets positioned right here, held in place by a magnesium right here. And that only happens when the bend occurs. Without the bend, there's no pocket for that holding to occur. So when the proper substrate has bound, the water's in place, the magnesium is in place, the proton can get removed, and the nucleophile can be created. Okay, so we've said a lot today about nucleophiles. Once that nucleophile is created, it attacks the phosphodiester bond. The bond gets broken, just like we saw with the peptide bond, and everybody's happy. Okay, I will very briefly go over that next time, and I will see you on Friday. Helicase is not a restriction enzyme. Helicase is a DNA unwinding protein, so it physically separates the strands, but it does not break any bonds.
Okay, so whereas I guess restriction enzyme is...
A restriction enzyme is breaking bonds.
Okay.
Yeah. So if you take a helicase and you treat DNA with it, what you have are two double strands that become two single strands.
Right.
The one double strand has two single strands.
Here, like,
Right.
But
if I take a restriction enzyme and I treat it, I will get, instead of one double-stranded piece, I'll get two double-stranded pieces.
I get it. Okay.
Okay? Sure. Hey, John, how you doing?
Hey, Kevin. Nice hat.
Thank you. I like yours, too.
That time of year.
It is. Well, no, summer. Some of us have bald heads. Summer's when we need those. You can tell the difference between a guy with hair and a guy with no hair here, right? Yep. Oh, hey, Emily. Excuse me. Yep, how you doing? Go ahead. Excuse me. Excuse me. How you doing, Veronica?
Good, how are you?
Good. How's your class going here? Good. Yeah, out jogging this morning. It was frost all over everything. It was pretty darn chilly.